Okay, so in this lecture, we'll go over the electron transport chain, and I also added ATP synthase, mainly because it just happens after the electron transport chain, and they're usually coupled together. Okay, so let's just go over a quick summary of how we get to this point. So we have glucose here. It's going in the blood, and it goes into the GLUT transporters, and then glucose enters the cytosol, and then we have our mitochondria here. So this is where TCA happens, and then this is where this happens, the electron transport chain. So glucose enters through glycolysis, and then we get electron carriers, and then when we also make pyruvate, pyruvate enters the TCA cycle, you make more electron carriers, and then the electron carriers are travel to um, go into the electron transport chain, and this is where we get most of our energy. So this is considered aerobic respiration because we require oxygen, so we need O2. So this is opposite of if we were to run glucose, pyruvate, the lactate. So this would be anaerobic. And then the cell does this when there is no oxygen. So we run aerobically if we have oxygen, and then if we don't have oxygen, we run anaerobically. So the electron transport chain is pretty much what aerobic respiration is. It occurs in the mitochondria, like I mentioned earlier, and the reason it was adapted was because it generates way more ATP per glucose molecule than anaerobic respiration. And when we'll talk about the mechanism of each complex, we'll see that oxygen acts as the last electron carrier, so that's why it requires oxygen. It's called aerobic respiration because it requires oxygen. Okay, let's talk about the mitochondria first because this is where the electron transport chain happens. So it has a double membrane, inner and an outer membrane. So the outer membrane is usually permeable to most things. The inner membrane is selectively permeable. It allows some things in and some things not. So this is the mitochondria here. I'm going to try my best to draw this. And then we have the inner membrane. So this would be outer membrane. And then we have the inner membrane here. Inner membrane. Inside, we'll just say it's the matrix now, but it like loops around like you see in this graph, and it's called like a crista shape. I don't know how to pronounce it, but inside is the matrix, and then between the outer membrane and inner membrane is the intermembrane space. So all the enzymes for the electron transportation are on this membrane right here, on the membrane between the matrix and the intermembrane space, which is the inner membrane of the mitochondria. If we go back here, so this is the inner membrane space, and inside here will be the matrix. So all our complexes, all of these, are in the inner membrane space. So the inner membrane mitochondrial inner mitochondrial membrane is made of mainly of this. It's made of a lot of lipids, and it has this cardiolipin um, lipid. It has this lipid called cardiolipin. This cardiolipin is made of a glycerol molecule. So this is a glycerol, and then we have four acyl chains. So this is one, two, three, four. So we have four acyl chains, and then we also have phosphate groups, and then this gives it a negative charge. And we'll see why this is important later when we talk about the proton gradient that we make. So this gives it a negative charge. Okay, let me zoom down more so we see the bottom. Okay, so specific interactions with oxidative phosphorylation. One of the cardiolipin's purposes, it interacts with a lot of the enzymes in oxidative phosphorylation, and it also provides stability when it traps the protons, and we'll see why that's important later on. Okay, so this is the cardiolipin composition. I don't think you guys are expected to know that. I just added it for extra info. So like I said, it's made up of 4 acyl groups, and then these are the different types that you can have, and then you can have different ones in the heart, and you can also have different ones in the liver. But it's pretty important for the electron transport chain because it stabilizes a lot of the protein uh, and and the proton gradient that we will make and we'll talk about. Okay, so some specific things that it does. It anchors. This is supposed to say creatine. So it anchors creatine kinase and nucleoside diphosphate kinase into the membrane. This is involved in transferring like ATP, mainly ADP. Uh, it's also involved in apoptosis. It releases cytochrome C into the cytosol. When cytochrome C into released in cytosol, if you guys are taking Synthase, Dr. Launderville will go over this. So when cytochrome C is released into the cytosol, usually that's not a good thing because cytochrome C is in electron transport chain. When it's released into the cytosol, this usually causes apoptosis in the cell, and then you have cell death. And then heart failure and diabetes have been associated with changes in the cardiolipin composition. So it's pretty important, these compositions here, for stability and function of the electron transport chain, because it makes up most of the inner membrane space of the mitochondria. Okay, so let's look at some enzymes linked with the electron transport chain. So remember in TCA we talked about succinate dehydrogenase. So that is usually coupled with complex 2. So succinate dehydrogenase, so you have the succinate, and then you go into succinate dehydrogenase, and then you make fumarate. And this reaction over here produces FADH2, and that can actually enter complex 2 and function in electron transport chain. So that's one way it's linked electron transport chain. When we go over fatty acid oxidation, we'll see that fatty acid oxidation happens actually inside the matrix. So most of the enzymes in fatty acid oxidation are also linked to ETC activity. And then perforance synthesis runs in the matrix, and it's also linked in ETC. This is the synthesis of like heme groups and stuff like that. That's what it means by perforance synthesis. It also occurs in the matrix, and it's linked to ETC. So you can see anything that has something to do with the matrix will probably be linked to the electron transport chain. Okay, so how it works is, so we make these, I think I have it later. No, I don't think so. So, actually I do. We make a bunch of NADH and FADH2 from running glycolysis, from running TCA. Some of the shuttles that we run, malate aspartate shuttle, glycerol 3-phosphate shuttles. So we make these, and we pretty much, the reduced carriers, that means they carry electrons. And when they transfer electrons to other cofactors in the electron transport chain, we use that energy that is generated by the transfer of electrons, and we pump protons across from the matrix into the intermembrane space, and we create this proton gradient, and then eventually we use that electrochemical gradient that we created, and then we generate ATP via ATP synthase over here. So that's general overview of what the electron transport chain is. So it's a series of oxidation and reduction reactions. So I have a brief summaries of what oxidation reduction is, if you forgot that from general chemistry. So the electron transfer is what actually allows us to create the proton gradient, and then that proton gradient is then used to generate ATP via ATP synthase. So before we talk about electron potential, redox potential, so this is what it's usually stated in. You'll get a half reaction, and it'll give you the redox potential of it. And the redox potential is how easily a compound can get reduced. So the more positive the value, the more likely it will be reduced. And the more negative, the more likely it will be oxidized. So this is if you're given redox potential, which is what most books and what most tables do. If you see this is what redox potential is. If you see oxidation potential, it will be different than reduction potential. So just to keep in mind, the more positive the reduction potential is, the more likely something will be reduced. Okay. So the way they measure it is they run this cell, two cells, and then they compare them to a standard reference. Usually they use a hydrogen electrode. They set it at 0 volts and then pH 7, and they measure what's called electromotive force and the amount of voltage that's created by passage of electrons compared to the reference cell is what the reduction potential of the specific reaction we're looking at is. It's a little confusing. If you don't understand that fully, just know that reduction potential is the more positive it is, the more positive it is, the more likely something will be reduced. So let's look at some examples. So like I said, the more positive, more likely to be reduced. If this standard reference thing is confusing you guys. So the more positive, the more likely it will be reduced, and the more negative, the more likely it will be oxidized. So here we have a couple of I think this is from the book, of different things in the reduction potential. So we see oxygen here as the highest oxygen potential out of everything in electron transport chain. That means it's the most likely thing to be reduced. That's why it's the last electron carrier. Electron transport chain just transfers electrons from things with low redox potential to eventually oxygen, which has the highest reduction potential. And this allows us to, without the use of any energy, to transfer the electrons all the way from NADH or FADH2 to oxygen. So we'll see that succinate has a really negative, NAD is negative, FAD is negative. And then once we get to like cytochrome C, which is in complex 3 and 4, it starts getting more positive. So toward the end of the electron transport chain, all the cofactors there will have a more positive reduction potential. The last things at the end of the ETC will be more likely to be reduced. That's why they have a higher reduction potential. And then from this table, we can actually predict electron flow. And the modification of electron potential is actually dependent on the environment. You'll see here that FAD has two different reduction potentials. Right there, it has two different reduction potentials, and that's because this is as a free coenzyme, and this is in a flavoprotein. So the environment can also affect the reduction potential of something. Okay, so let's look at an example here. So we have zinc 2+. We add 2 electrons, and then we get zinc S. This is just general chemistry review. If anyone forgot it, I just added in case anyone forgot it, because this is the basis of everything in the electron transport chain. And then we have copper plus 2 electrons, and then we get solid metal copper. So we'll see this one has the more positive, so this is the one that will be reduced, and this is the more negative, so this will be the one that's oxidized. So in order for this to be reduced, it accepts electrons. So we'll leave it as it is, because the reaction given is the one that it's accepting electrons. And since this is the one that's going to be oxidized, we just flip that one. So flip this one, because that's the one that's going to be oxidized. And then we get zinc solid. And then that gets oxidized to zinc 2+ plus 2e electrons. And then usually you would balance the electrons, but because we have 2 electrons here and 2 electrons here, they add up. So then we add the zinc with the copper 2+, and then we add this Zn2+, and then the copper solid, and that's how we get our final reaction. So I know this sounds really simple, but in case someone forgot it, I just add added it in case just you need a quick review on it. So this is our final reaction. Okay, that's a lot of errors. So with this reduction potential. Oh, by the way, when we calculate the reduction potential, we just we add these. We flip whenever we flip it, we make it positive, and then we add them. So this would end up being like what 1.10 because we flip this one, so this one's positive, and then we add them. And then some people do it. You subtract the negative. As long as you get the same value, it doesn't matter how you do it. Okay. So from the delta E, which is the reduction potential, we can calculate our delta G, which tells us how spontaneous something is. So this is called the Nernst equation. I'm sure you've seen this before. N is the number of electrons transferred. This is Faraday's constant, which is, you can just use, 1 second. We can just use 96,500 joules per volt or 96,500 coulombs. Joules per volt is the same thing as 1 coulomb. And then, yeah, like I said, basically, the free energy can be calculated if we know the reduction potential. Okay, so then I also added this here. This equation comes from the one that relates E with K. And it has natural log in it. And then you get this 2.3 when you convert from log to natural log. And then we have the RT and F, and then natural log of products over reactants. And then this can also allow us to calculate E from the equilibrium constant, pretty much. I don't think you were expected to know this one, but I just added it because you can also relate E with K. The equilibrium constant. Okay. So before we'll start anything, let's look at the overview of before we start going over the complexes. So the electron transport chain, we start with NAD and NAD. We start with NADH, and then we give the electrons to oxygen, and then oxygen becomes water. So if we were to calculate the free energy, if it were to run perfectly efficient, we can calculate that by the difference in voltage and use the Nernst equation. So we would flip this one and then add them or subtract the negative. So we'd have 0.82 plus 0.32 volts, and then we get 1.14 volts. Faraday's constant is 96.5 kilojoules per volt. And then the number of electrons we transfer between NADH and oxygen is 2 electrons. So this is the delta G of the electron transport chain. So you can see this is very negative. So this is the theoretical value. We'll go over the actual value at the end. The theoretical value is very negative. That means, which gives us plenty of energy to synthesize ATP. See, it's exergonic, and then it synthesizes a lot of ATP. Okay, so when we go from glucose, this is how we get all our electron carriers. To pyruvate, we make 2 NADH from gap DH when it splits, and then because we have 2 gaps, we'll end up making 2 NADHs. This is supposed to be C2 down here. And then we make 1 NADH from the pyruvate dehydrogenase complex. We make 1 NADH in isocitrate dehydrogenase. We make 1 NADH from alpha-ketoglutarate dehydrogenase, and then 1 FADH2 from succinate dehydrogenase. And then we make our last NADH from malatoxyl acetate. So the ones from the pyruvate dehydrogenase complex in TCA are already in the mitochondria. So those ones don't really need to do much to travel to get to the electron transport chain, because TCA is in the mitochondria and the pyruvate dehydrogenase complex is in the mitochondria. So those ones can just go directly into the electron transport chain and then generate even more energy for the cell. But for the NADH we make in glycolysis, those can enter. If you guys go back and go over the malate aspartate shuttle, or the malate shuttle, or the glycerol 3-phosphate shuttle, you'll see that this NADH can enter the mitochondria. through malate aspartate shuttle or glycerol 3-phosphate shuttles. So it can enter the NADH that we make from glycolysis can enter the electron transport chains through those two shuttles. Okay, so basically, like I said, what we're doing is we're oxidizing and reducing cofactors through a stepwise transfer of electrons, and then eventually we get the oxygen. And then this is just a theory, but is that when we remove electrons from coenzymes, this is actually coupled to pumping, which is what we learn in biochemistry and what we learn in textbooks. It's actually just a theory that when we transfer electrons between the different complexes, this is coupled with pumping protons across the matrix. This diagram should be this way. From the matrix into the intermembrane space. We pump protons from the matrix into the intermembrane space, and then that eventually gives us the proton gradient to make ATP. Thing to note is complex 2 does not pump protons, which we'll talk about when we go over the complexes in detail. Okay, so I think we can, no wait, I have one more slide before we can talk about complex 1. Okay, so this is the reduction potential, so this is the negative side, and this is more positive. So more positive will be more likely to be reduced, and then this is more likely to be oxidized. So we'll see that oxygen is down here with H2O, and then we'll have cytochrome A3, and then cytochrome C is here. So this is in complex 4. And then we also have NAD and NADH up here, so those are in complex 1. So we'll see that, not 4, complex 1. So we'll see that as we go into the electron transport chain, the thing that we're giving electrons to have a more positive reduction potential because they need to somehow be the thing that's getting reduced. So that's why oxygen has the highest reduction potential, and then NAD and NADH, which is what started the electron transport chain, has the lowest reduction potential. So like I also listed the values here. Oxygen to cytochrome A3 has a so this is the last reaction. Oxygen to cytochrome A3 has a really positive reduction potential, and then NAD to NADH2 has a really negative reduction potential. Okay, I also mentioned, so these are covalently attached to complexes, so these are iron sulfur centers that help in electron transport chain. And then FMN is also in complex 1. And then we'll talk about coenzyme Q, with another name for it is ubiquinone. Ubiquinone or ubiquinol. Ubiquinol is the reduced form of ubiquinone. And then cytochrome B, cytochrome C. There's a bunch of different cytochromes that you guys have to know, unfortunately, that we'll go over in details. But as we go into the electron transport chain, we have a more positive reduction potential in our cofactors. Okay, now we can actually start talking about sorry though, it's too long, about what's going on in the electron transport chain. So we have our first complex is complex 1. Sometimes it's called NADH ubiquinone oxidoreductase. It's made up of 45 subunits. That's a really big protein. 100 kilodaltons and it has 9 redox cofactors. The bacteria version of this is still even really big. It has 14 subunits. What we do in complex 1 is we transfer electrons from NADH to coenzyme Q, and then when we do that, we pump 4 protons. It's important, 4 protons across the membrane. So we go from NADH to coenzyme Q, and that's coupled with 4 protons being pumped across the membrane. Like I said, another name for coenzyme Q is ubiquinone. That's why it's in the name. So ubiquinone is the oxidized form, and then ubiquinol is the reduced form. So you might see both as when they're referring to coenzyme Q. Okay, and then I added here that coenzyme Q has this. I'm trying to zoom in. Has an isopropyl tails, and it allows it to stick in the mitochondrial membrane and shuttles back and forth between complexes. Because coenzyme Q takes electrons from complex 1 and then brings those electrons to complex 3, and it also takes electrons from complex 2, which we'll talk about, and then gives the electrons into complex 3 again. So it needs those isopropyl tails to anchor it in the mitochondrial membrane, and then allows it to shuttle back and forth between the complexes. Okay, let's look at the mechanisms of the electrons transferred in complex 1. So we have our NADH, so this comes from the shuttles. It can come from TCA, even lactate dehydrogenase can produce the electron carriers, the electron carrier NADH. So this can come from many things. So NADH gives 2 electrons to this cofactor called flavin mononucleotide, or you can just call it FMN. And then FMN gives the electrons to iron sulfur clusters, and then iron sulfur clusters gives the electrons to coenzyme Q. So why can't we just go and give electrons from NADH to coenzyme Q? So NADH has 2 electrons, and it can only give 2 electrons at a time. But the iron sulfurs, which is right before coenzyme Q, accepts 1 electron at a time. So we can't just give the electrons to the iron sulfurs or coenzyme Q right away, because both accept 1 electron at a time. So we need this. We have this intermediate called FMN, and that kind of helps take the electrons from NADH 2 at a time, and then give it to iron sulfur clusters 1 at a time. So that's why we need all these cofactors in the middle, because some things can only accept 1 electron at a time, and some things can only give 2 electrons at a time. So both complex 1 and 2, like I said, give their electrons to coenzyme Q. So when coenzyme Q gets its electrons, it's reduced into reduced form, which is called ubiquinol. This is the oxidized form. This is the reduced. And then this one is called ubiquinin. Okay, so I added that because I got confused a lot with it when I was first learning the electron transport chain. Also, ubiquinin is different from ubiquitin. Ubiquitin is that thing that tags proteins in the cell to be degraded. Ubiquinin is the one in the electron transport chain. So don't confuse these two, especially if you're taking cell phys this semester. Okay, so I also added FMN down here. So this is our flavin mononucleotide. So this is the oxidized form, and then this is the reduced form. You can see it accepts 2 electrons, and it does that. It's our mediator in the middle because it can take 1 electron at a time. Because NADH can give 2 electrons, can only give 2 electrons at a time, but FMN can accept either 1 or 2, and then it gives the iron sulfur clusters from this semiquinone. So this gives it the iron sulfur down here, because iron sulfur can only take 1 electron at a time. So we have this FMN in the middle because it can hold 1 or 2 electrons depending on the things before it and after it. Okay, so now that was complex 1, we had our electrons in NADH, and we give those to coenzyme Q. Now complex 2 is called succinate ubiquinin oxidoreductase, and it's also coupled with succinate dehydrogenase. So we'll see complex 2 is like functions on this side, and then succinate dehydrogenase is inside the matrix, and it helps function in as our TCA enzyme. So complex 2 has 3 iron sulfur centers, very similar to the ones in complex 1. And it's 70 kilodalton subunit, and it contains an FADA that is covalently bound to a histidine residue. Okay, let's look at the mechanism of complex 2. So like I said before, complex 2 also gives the electrons to coenzyme Q. So both complex 1 and 2 give their electrons to coenzyme Q. So this is our mechanism, and it doesn't have to enter through succinate. It can, I have other ways where it can enter. I'm glad I added that slide. So succinate and FAD, we get our fumarate and FADH2. So this is our TCA reaction. If you go back and review TCA, this is the same thing. You have succinate, FAD, and then you give the FAD gets reduced, and then succinate gets the oxidized to fumarate, and then FADH2, and then oxidized coenzyme Q. The electrons from FADH2 are given the oxidized coenzyme Q, and then we get reduced coenzyme Q. I'm going to call it CoQ. Actually, no, it's not CoQ. I don't want to confuse anyone. And then we get back our FAD, which can go back and help in the succinate dehydrogenase reaction. So something important, like I said, is there's no proton pumping in this reaction. So this reaction does not contribute, this complex does not contribute to the proton gradient. It's mainly just a way for FADH to enter the electron transport chain, FADH2. So we'll see that equilibrium is approximately around 0, and then voltage is, the delta E is slightly above 0. Okay, so let's look at other ways that things are sent into the electron transport chain. So I mentioned before that the electron carriers made in the mitochondria can directly enter the electron transport chain because everything is in the mitochondria, so it's really easy for the electron transport chain to take those electron carriers and use them. But for example, glycolysis makes the electron carriers in the cytosol, so we need to somehow transfer those electrons for the electron transport chain to use. So if you remember in glycolysis, I talked about the glycerol 3-phosphate shuttle. It takes this dihydroxyacetone phosphate, this is DAP, and then we run this shuttle where we give the electrons to glycerol 3-phosphate, and then we generate NAD. So this can go back and help in GAPDH in the cytosol to help run glycolysis faster. And then the glycerol 3-phosphate can then give the electron back to DAB, and then glycerol 3-phosphate can get oxidized back to DAB, and it gives the electrons to FAD, and it becomes FADH2. And then this can then enter complex 2 and give the electrons to coenzyme Q, and then you'll get reduced coenzyme Q. Does that make sense? So this is one way that the electron carriers we made in glycolysis enter into the electron transport chain. So this shuttle helps us make NAD plus for glycolysis to run faster, and we transfer those electrons to be used in the electron transport chain. Another way it can be used is in beta oxidation. Beta oxidation for now, it just happens in the matrix. And then when there's this reaction done by acyl-CoA dehydrogenase, we take acyl-CoA and then we make this eno-CoA, where we have this double bond over here. And then when we reduce a single bond to this double bond, it gets oxidized. So then the electrons, it gets oxidized, so then FAD gets reduced to FADH2 because those electrons need to go somewhere. So FAD is the one that accepts it, and because this happens in the matrix, FADH2 can go directly into ETC. Okay. We move on. So now we have complex 3. So this is probably going to be the most complicated one out of all of them, and I'll show you why in a second. So it's called ubiquinol. You can also call it ubiquinol cytochrome C oxidoreductase. It has 11 subunits, and it has 3 prosthetic groups that are redox centers. Those are actually important. One is cytochrome B. Cytochrome B has 2 heme groups. We have cytochrome C1. So cytochrome C1 is different from cytochrome C, and don't get that confused. There's a lot of cytochromes going on. So cytochrome B, we have cytochrome C1, and then we have this iron sulfur protein. It's called Reisky iron sulfur protein. And it has 2 iron sulfur centers in there. And cytochrome C1 has 1 heme group. So the whole point of complex 3 is to transfer electrons from ubiquinol or reduced coenzyme Q to cytochrome C. Okay, this cytochrome C is different from cytochrome C1. So this is a prosthetic group in complex 3. The electrons we're transferring are to a cofactor called cytochrome C. 2 different things. Okay, so let's talk about the different hemes. So if you took Biochem 1, you'll go over like hemoglobin. This is the one used in hemoglobin. So there's different types of hemes. So you have heme A, heme B, and then heme C. So there's a couple of heme Bs here. I don't think there's heme. There might be a couple heme A's and heme C's in somewhere. I forgot what protein uses what hemes. But basically the hemes are different based on the group attached to them. So heme B over here, so this is the one used in hemoglobin. My pen right now. So heme B is used in hemoglobin, and then heme A and then heme C have different groups attached to them. The oxidized form for all of these is whenever the iron has a +3 charge, and then the reduced form whenever the iron has a +2 charge. The reason these hemes don't bind oxygen like they do in hemoglobin is because they're embedded inside the protein, and that's why they don't bind oxygen in electron transport chain. Otherwise they would mess up everything if they're just binding the oxygen and then the oxygen is not being used as an electron carrier. Okay, so complex 3 has this thing called the Q cycle, and you'll see why this is important. So we have this reduced coenzyme Q, and it has 2 electrons, but we have a problem. Cytochrome C can only take 1 at a time. So this thing wouldn't exist if cytochrome C can take 2 electrons at a time, but because it decides it wants to take 1 electron at a time, we have to go through this whole Q cycle, which is complicated. But hopefully I'll try to explain it as easily as I can. So let's go back. Let's first identify the what is it? Here, let's zoom in. Let's first identify the 3 prosthetic groups that we talked about. So we talked about cytochrome B as one prosthetic group, and then we said cytochrome C1, and then this is our iron sulfur Reisky. So we have this reduced coenzyme Q. So this comes from complex 1 and complex 2. So it can come from either. It enters cytochrome B. So that's the first thing it does. And what it does is it gives 1 electron to iron sulfurs. Remember, we talked about in complex 1, the iron sulfurs can also only take 1 electron at a time. So it gives 1 electron to iron sulfur up here, and then 1 electron goes down. And when it gives 1 electron to iron sulfur and 1 electron to, it's called heme Bl, it pumps 2 protons across the membrane. So 2 protons from the matrix go into the intermembrane space. The iron sulfurs, they have the 1 electron. They give that to cytochrome C1, and cytochrome C1 has a heme C1 in there that accepts the electrons, and then cytochrome C1 gives the electrons to cytochrome C, gives 1 electron to cytochrome C. So we want 1 electron to iron sulfur, and then we want the cytochrome C1, and then we want the cytochrome C. And then cytochrome C can translocate and then go translocate, and then goes into complex 4. So now we have one cytochrome C with one electron, but we need to transfer the other electron somehow. So remember we said we gave one electron to iron sulfur, and then the other electron is given to this heme Bl. So this is the low potential heme, and then one electron is given from a low potential heme to a high potential heme, and then we have this oxidized coenzyme Q. So that would be an oxidized coenzyme Q from previous cycles, and then that coenzyme Q accepts the electron from the high potential hemes, and then we get this semi-quinone, I think that's how you pronounce it, which a coenzyme Q with only one electron. So coenzyme Q can bind either QP when it's in heme Bl, or it can bind at Qn when it's at heme BH, which is the low potential heme and the high potential heme respectively. Okay, so we did all of that and we transferred one electron to cytochrome C. So we have this oxidized coenzyme Q that goes in the membrane of the cell. So it's 2 electrons, it gave one down here and then one up here, right? And then this coenzyme Q can go accept 2 more electrons from either complex 1 or complex 2, and we get this reduced form of coenzyme Q. And then this reduced coenzyme Q does the same thing. It gives 1 electron up here to iron sulfur, and then it gives 1 electron. The iron sulfur gives the electron to cytochrome C1, and then cytochrome C1 gives the electrons to cytochrome C. And when it does that, it pumps 2 more protons, so total we have 4 protons pumped. And then the remaining electron travels down the low potential heme and then high potential heme. And then we get this semi-quinone, and then this can grab protons from the matrix and then become the reduced form, and then this can go and start the cycle again. Okay, so another way to think about this semi-quinone that we left. So this semi-quinone can also grab an electron here. So the semi-quinone can grab this is the same thing as that. Let's think about it like that. So the semi-quinone can grab the remaining electron downward, and then it becomes the reduced coenzyme Q, which can start the reaction again. Because you'll see that we used 4 electrons. We used 2 here, and then 2 here, right? So one goes to cytochrome C, the other goes to the other cytochrome C, and then the remaining 2 goes to these semi-quinones, which eventually become 1 reduced coenzyme Q. Okay, I'll say that again. So we used 4 electrons from coenzyme. We used 2 reduced coenzyme Qs. Each one has 2 electrons. Each one gives 1, so that would be 4 electrons total. So each one gives 1 electron to cytochrome C. So we'll have 2 reduced cytochrome Cs. And then the other remaining 2 electrons, each one goes to, they get added to oxidized coenzyme Q one at a time, and then you'll eventually get a reduced coenzyme Q that can start the reaction again. So two of our electrons are transferred, and the other 2 just start the reaction again. So that's why we technically use like 4 electrons, but we're only transferring 2 electrons. Okay, if you're confused about this, you can try to read the book, or you can ask me questions, or you can try to rewatch this and see if you understand it better. But I'm gonna erase all of this writing that I put down. Okay, but yeah, this is very important, and I guarantee there will be like some question on this, because it's just complicated, and professors like to ask about stuff that's complicated. Okay, so like I said, cytochrome C shuttles back and forth between complex 3 and complex 4. This is complex 3, and then it travels to complex 4 and gives the electrons to complex 4, which is our last step. Okay, cytochrome 4, it's called cytochrome C oxidase. It has 13 subunits in mammals, and in bacteria it has 3 to 4 subunits. The whole point of it is it transfers electrons from cytochrome C to oxygen, and then this is also coupled with movement of protons. So we moved protons in complex 1, and then we moved protons in complex 3, and now we're moving protons in complex 4. The only complex that we don't pump protons in is complex 2. Okay, the 3 largest subunits are encoded by mitochondrial DNA, and the rest is encoded by nuclear DNA. Nuclear DNA is what's inside the nucleus. Mitochondrial DNA, you get that from your mom. So you inherit that from your mom, and it's DNA inside the mitochondria, and it can actually code for a bunch of stuff. Okay, so the 3 largest subunits are subunit 1, subunit 2, and then subunit 3. Subunit 1 is a transmembrane domain. That means it's in the membrane. It has a heme groups A and A3, and then they're bound to a protein called CUB. Subunit 2, it has the binding site for cytochrome C, which is a protein called CUA. And this large subunit actually extends from the cytosolic phase into the inner membrane space. And then we have subunit 3, which is a 7 transmembrane helicase, and then those are the ones that transfer protons to contribute to the gradient. Okay, so let's look at the mechanism of complex 4. So we have a reduced covalent cytochrome C, so we have 2 of these, because remember, when we do the Q cycle, we can only give 1 electron at a time to cytochrome C. Those cytochrome Cs give it the protein CUA, and then CUA gives it to heme A, and then heme A gives it CU, this is supposed to be a plus sign, it gives it a protein called CUB, and then heme A3, and then when you do that, you pump 2 protons across the membrane. And then because we have 2 cytochrome Cs going through this, we're technically pumping 4 protons. So we pumped 4 protons in each of the complex 1, complex 3, and complex 4. We don't pump protons in complex 2. This is the mechanism. So this is cytochrome C. And it gives it CUA, and then that gives it A, and then gives it A3. And then cytochrome B, and then cytochrome B finally gives it to oxygen. And then oxygen accepts the electrons and then becomes water. And then this is the end of the last electron carriers. It's not the end of the lecture, there's still a bunch of stuff. Okay, so when we transfer the electrons, we make this proton gradient. So we have this, we make this intermembrane space become more positive, and then the matrix becomes very negative, because all the complexes pump protons in this direction, so this intermembrane space becomes more acidic from all the protons being pumped, and then the matrix ends up becoming more basic or alkaline. So remember, I talked about cardiolipins at the beginning, and I said they have the negative charge in their phosphate groups. Those actually help stabilize those acidic protons in the inner membrane space. So that's one reason one of the examples cardiolipins contributes to stabilization of the proton gradient. So we make a voltage difference, and then this is also a change in pH. So the voltage difference is from the positive sign in the intermembrane space and the negative sign in the matrix, and the change in pH is from those being protons, and then those contribute to pH. So this is an electrochemical gradient, and that's actually enough that allows us to synthesize ATP, which we'll talk about ATP synthase in a bit. Okay, so overview before we start ATP synthase is that we went from NADH to oxygen. Approximately 10 to 12 protons are pumped, and then our free energy, when we use this equation over here, this is also derived from the delta G equals negative RT natural log of K. but then K is changed into pH and voltage. And then instead of natural log, they do a bunch of algebra, and it ends up being 2.3. The pH changes from 0.71 to 1 pH unit. Voltage changes from 1.5 to 2 volts. And then we end up, when we calculate it, we'll get around 200 kilojoules. Remember we did the calculation at the beginning and we got 219 kilojoules. So this is very close. So electron transport chain is actually very efficient in terms of conserving the energy when it transfers the protons across the different complexes. So it's a very efficient system. Okay, so there was an experiment that this is how they found that oxygen is used as an electron carrier, and then it goes into respiratory control as well. And it allows us to conclude that ETC, the electron transport chain, and ATP synthase are coupled together. So what they did is they added mitochondria in a buffer, and then they gave it succinate, and then they tracked the oxygen consumption over time. Okay, and what they found is that when the ADP was around 0, if ADP is around 0, that means ATP is high. The mitochondria started consuming very low oxygen. Okay, so they when they were like first figuring this out, they were like probably curious. So we start to consume a lot of oxygen for a certain time until we run out of ADP. So what they ended up concluding is the activity of the gradient that we produced is dependent on ATP levels, and then that those ATP levels can actually regulate how ETC is ranged. So when ATP levels are really high, when ADP is low, the electron transport chain won't run as fast because we have a lot of energy. That's why it's not consuming oxygen. So from this experiment, they were able to conclude that ETC and ATP synthesis are coupled together. So in healthy mitochondria, pretty much they can't occur separately. They usually occur together. They're two different things, but they occur together. Okay, so there's this phosphate-oxygen ratio. This is a way to calculate how many. I just included this because I thought it was helpful, and I think Dr. Shriver went over it. Basically, it's a way to calculate how many ATPs we produce per molecule, pretty much. So it's the way it's defined is the maximum number of ATP molecules made as an electron passes down the respiratory chain from substrate to oxygen. So that's the exact definition of what a PO ratio is. So let's look at NADH, for example. So when NADH goes in, it pumps around 10 protons, and then it transfers 2 electrons from NADH to oxygen. And then 1 molecule of ATP synthesis requires 4 protons. When we go over ATP synthase in a bit, you'll see why it requires 4 protons. So from this, we can calculate the PO ratio. So we have 1 ATP per 4 protons pumped. If we multiply that by, so we pump 10 protons per 2 electrons, and then per 1 NADH molecule, we had 2 electrons. So if we do the math and cancel this out, we'll end up with the 2's cancel, and then we'll end up 10 divided by 4, and we'll get 10 ATP molecules per 1 NADH. So if you ever see those values in the book, that's how they calculated them. They calculated them with the PO ratio. So succinate has a PO ratio of actually 1.5, same as FADH2, and the reason is because they start at complex 2. They bypass complex 1, so they're not pumping as much protons as 1 NADH molecules. That's why NADH produces more ATP than FADH2 or succinate. And the reason this ratio works, it's called the PO ratio, is because the amount of phosphate that is consumed when we make ATP synthesis is for what the P stands for phosphate and oxygen, O stands for oxygen, and it just basically relates the amount of phosphate that is consumed for each oxygen molecule that is reduced to water. That's all what the P is and what the O is in the PO ratio. Okay, so now we made past all those electron carriers. We passed them from complex 1 to complex 4, and then we created this proton gradient. So we need to somehow make ATP out of that proton gradient. So we have this thing called ATP synthase. Sometimes it's called complex 5. If you ever see that, complex 5, they're just referring to ATP synthase. So it's around 400 to 500 kilodaltons. It's in the same membrane as all the electron transport chain complexes. So it's an inner mitochondrial membrane. And it has two domains. So it has an F1 and F0 domain. The F1 domain acts as a binding site for ATP, and it also catalyzes ATP synthesis. The F0 domain is basically a channel for protons. So this is the F0, and this is the F1. And then remember I said 1 molecule of ATP synthesis needs 4 protons. This is what I mean by that. 3 protons are needed for the synthesis of 1 ATP molecule, and then we need 1 proton for ATP transport in exchange to ADP and P. And we'll see. So this is the 1 proton they're referring to when it enters the F0 molecule, and then the 3 function in F1 to make the ATP molecule. And then I have the mechanism for all of that, so we'll go over it in more detail. Okay, so just go over the structure of it because it can be confusing. So F1 has alpha, beta, gamma, delta, and sigma. It has 3 alphas, 3 betas, 1 gamma, 1 delta, and then 1 sigma. And then our F0, for some reason, decided to name them in English for the F0 or F naught. They're called A, B, and C, and then there's 2 A's, 2 B's, and then around 10 to 12 C's. Okay, let's go over F0. So this is, it can be a little complicated, but I'll try to explain it. So this is the channel for the proton. So this is the proton gradient that we made. So this is the inner membrane space. This is the matrix. And we have all these protons in the matrix. So this is subunit C in F0. And then it has those 2 half channels. So it has a half channel in the facing the intermembrane space. And it also has another half channel facing the matrix. Okay? So what happens is, let me get the pen. So a proton moves from the intermembrane space into a half channel. And what it does is protonates an aspartic acid residue, and it's at position 61. So this is a negatively charged aspartic acid residue. We have a proton that comes in. Let's see the arrow being drawn there. And then it protonates the aspartic acid residue. Okay? Once it does that, it creates, it allows the channels to rotate clockwise. So you'll see here it's rotating clockwise. And then the half channels in C rotate clockwise. And then we expose a previously protonated aspartic acid residue in a half channel that's now facing the matrix. When we rotate, this half channel opens to the matrix, and then the aspartic acid residue in there gets deprotonated, and it sends the proton into the matrix. Okay, so a proton comes from the intermembrane space, protonates aspartic acid residue in the half channel facing the intermembrane space in subunit C. This causes a rotation, and when we rotate, we expose a previously protonated aspartic acid residue to face the matrix side, and then it sends a proton in the matrix. Okay, this process is repeated, and it rotates complex. It rotates subunit C, and then this torque is actually read out in F1, which is eventually allows conformational changes, which allow for ATP synthesis. And it's also coordinated by an arginine residue, but it's not a specific detail that you guys need to know about that. Okay, so if you're confused about this, I would go back and replay it, or just read the book about it, or watch a different video if you don't think I explain things pretty well. Okay. Oh, another thing I'll mention is the spinning with subunit C is transmitted through F1 via the sigma and the delta subunits. So this is the sigma, and this is the, I think those are the right letters. No, this is sigma and gamma, not delta. Sigma and gamma. So when subunit C rotates, so this is subunit C, when it starts rotating, this torque is transmitted into F1 via sigma and gamma. Yep, sigma and gamma. I don't know where I got delta from. Okay, so now that we rotated this, we're going to induce the conformational change in the beta subunits in F1. And a thing to know about this is that delta G is actually near zero. So the synthesis does not require energy. So when we make ADP into ATP, this does not require energy. Delta G is equilibrium. What requires energies is the binding and I binding of ATP to the F1 complex. So it's a very little difference, but it's important to know. The delta G for ATP synthesis is around zero, but we still require energy via the proton gradient. Okay, so now we have protons from the intermembrane. We have protons from the intermembrane space going inside the matrix, and then we create this torque, and it allows the spinning of the beta subunits of F1. So F1, the beta subunits, so let's zoom in on this, just to briefly go over it. So we have B0, not B0, B not BL and BT. So those are different states that the beta subunit can be. The one in the middle here this is the gamma, and then it's coordinated by the 3 alphas. Okay, so the beta can exist in 3 different states: the O and the L and the T, or the not L and the T. So the O state changes to L, and then L changes to T, T changes to O, and then O changes to L, and then it just continues like that. Whenever the O state changes to the L state, it does that when it binds ADP and P. So whenever ADP and P bind to a beta subunit, it changes from an O state to an L state. And then the L state takes the ADP and P and makes ATP synthesis, makes ATP, and then when it creates ATP, it goes through a conformational change and becomes the T conformational subunit. Okay, so when we go from L to T, we make ATP. Okay, and then when the T changes to the O, we release the ATP. And then we start again with an empty subunit, the O subunit. And then once it binds to ADP and P, it becomes L again, and this just starts spinning, and we're just pumping ATP for all the the protons that enter the F naught subunit and allow us to rotate the F1 subunit. So the rotation is actually coupled with the conformational change. So we're rotating and changing from O to L to T. So not only are we rotating, we're also changing from O, L, and T at the same time. And it's also irreversible. This is hard to think about when you're keeping track of the O and L and T, because they switch to the next one and then it also rotates. For now, just know the 3 like sites and what each of them do, but I don't think you'll be expected to like draw a picture of this, because it can get really complicated. Okay, so the ATP that we make when we go from L to T, we make the ATP. This ATP must be released before we can start make another ATP, and then once it's released, we're back into the O state. ATP synthesis is maintained by adenine nucleotide transporter. So this transfers uh, ADP, ATP, and then it maintains ADP levels sufficient enough to be able to generate ATP for the cell. Okay, so if the gradient is flipped, remember I said it's irreversible. The reaction is irreversible, but if the gradient is flipped, this starts running, instead of running clockwise, it starts running counterclockwise. And what this does is, instead of we make ATP, we start hydrolyzing ATP. So ATP synthesis runs in the opposite direction, and we start breaking down ATP into ADP and P. So this whole thing will just run backwards. ATP would bind, then ATP would change to ADP and P, and then ADP and P would be released. So if the proton gradient is, this is why it's important to maintain the proton gradient. If it's flipped, if it's higher in the matrix, ATP synthesis runs backward, and then we start hydrolyzing ATP. This happens when there is no oxygen, and this is what happens in hypoxia. You don't have an electron acceptor. That's why if you don't breathe for a while, I guess, or if the cell's not receiving oxygen in a long time, the cell will die because you're hydrolyzing ATP. You don't have the final electron acceptor to be able to make ATP synthesis run in the proper direction. Okay, so they did this experiment. So this kind of this allowed them to figure out that you need the proton gradient for ATP synthesis to run. The other one was for that they're coupled together. This one, they figured out that you need a proton pump for ATP synthesis and the electron transport chain to run. You need a proton pump to make ATP, pretty much. You need the proton gradient. So they put a bacterial proton pump in a liposome, and then they had this ATP synthase, and then they had a proton pump right there. And so whenever they expressed this proton pump, ATP synthesis was able to use those protons and create ATP. However, when they don't express the proton pump, you're unable to generate ATP, and if there's actually more hydrogens outside here, ATP synthesis actually started running the other direction. So that's how they figured out you need the protons, and you need the proton pump to be able to generate ATP. Okay, so let's talk about what ETC can do when we're under hypoxia. So we mentioned under hypoxia we won't have an electron acceptor, the gradient gets flipped, and then ATP synthase would run backwards. So the cell has some adaptations to that to try to help as much as possible. So because if it doesn't, the cell will just use all of the ATP in the cell, and it will just start burning through energy, and it uses ATP from other cells, and then it will just be really bad. So the cell needs a way to regulate this. So under hypoxia, there is this transcription factor called HIF1 alpha, and what this does is it pretty much shuts down the electron transport chain. So oxygen is happy accepting one electron at a time. And this can actually give superoxides. And when this happens, the reason oxygen can accept one electron at a time is that its two valence electrons have the same spin. So it doesn't need to accept two electrons at one time. So it can take one at a time. And this can make a superoxide. And this is the danger of reactive oxygen species. We talk about how dangerous they are and how they can cause cancer and stuff like that. So if we only accept one electron at a time, this doesn't fully reduce oxygen to water. And then because we don't have the electron acceptor, you start hydrolyzing ATP. So the cell has this HIF1 alpha, which allows it to shut this down, to not allow oxygen to do that, or under hypoxic condition, so that we're not hydrolyzing ATP. So H1F alpha shuts down ETC under hypoxic conditions. Okay. So under, I'll just read briefly through this. So under low oxygen levels, we need ATP, right? Because we don't have enough oxygen. So we want to run ETC because our ATP levels are low, right? So we need to somehow make ATP. So ETC wants to run. But under hypoxia, our still main source of energy to do anything in the cell is ATP. So we still need to make ATP somehow, but we don't have any electron acceptors because we're under hypoxia. But if we do that, because we don't have the final electron acceptor oxygen, instead of making more ATP, we'll actually start hydrolyzing ATP, which is even worse for the cell. So what happens is, under low oxygen conditions, we don't want ADP to stimulate ETC to run forward, because then we'll just start hydrolyzing ATP. And this disrupts the membrane potential because we don't have the oxygen to act as an electron acceptor, so that we don't have the gradient, and then ATP synthesis can work in reverse, and it ends up being worse than what we started at. So we can't run ETC, and we can't have ADP stimulate ETC. So what cells have is they have this inhibitory peptide called IF1. It binds and inhibits ATP synthase under low oxygen conditions. And when it binds and inhibits ATP synthase, it stops it from hydrolyzing ATP, because under hypoxic conditions, ATP synthase will start hydrolyzing ATP. So we need to somehow stop that, because ADP is stimulating ETC, and because we don't have the electron acceptor, we don't have the proton gradient, so ATP synthesis is running backwards. So we need to somehow stop that whole reaction from happening. So the cells have this inhibitory peptide IF1. It binds and inhibits ATP synthase, and that way ETC still runs under low oxygen conditions, but we're not hydrolyzing ATP. Okay. So IF1 continued. So what it does is IF become inhibitory peptide. What it does is it becomes active under acidic conditions. So this happens, for example, if you're exercising, right, anaerobically, you're like doing a sprint, and you're holding your breath. So you don't have oxygen. So you're running anaerobic respiration. And when you run anaerobic respiration, you make lactate, and you're under acidic, very acidic conditions. The cell is because it's making a lot of lactate. The cytosol starts becoming really acidic. So that's when IF becomes active, because whenever the cell becomes really acidic, it's another way to tell the cell that, hey, we don't have enough oxygen to be running the electron transport chain in ATP synthase. So it becomes active under acidic conditions. And what it does is it shuts down ATP synthase, and then this allows us to stop hydrolyzing ATP under hypoxic conditions. So an example for this would be like if someone were to like sprint and hold their breath, their cells are still working, but they don't have oxygen acting as an electron acceptor. And the way it does that is it produces these reactive oxygen species, which eventually can act as signaling molecule, and then this induces apoptosis in the cell, because before the cell starts hydrolyzing ATP and leading to much worse things, maybe even like cancer and stuff. So the cell just goes through apoptosis, it kills itself before it causes harm to other cells in the tissue. So reactive oxygen species, they activate NF-κB, and then this can induce cell death. Okay, so I think I have a couple more slides here. I know this is going a lot longer than I expected. Okay, So uncoupling of ETC and ATP synthase, and I think you guys have a couple questions on the homework. So I think I told a couple people that when you uncouple ETC and ATP synthase, you will have reduced oxygen consumption. ETC would run slower. That's actually wrong. I apologize if I told you this. If you're listening to this, if you can go back and switch your answer, that would probably be better. Because what happens is when we uncouple ETC and ATP synthase, ETC actually runs faster. And I'll explain that in a second. So we disrupt the proton gradient with an uncoupler. So we decouple ETC and ATP synthase. We're still shuttling, we're still creating a proton gradient from complex 1, complex 3, and complex 4. But because we have this uncoupler, it shuttles H plus across the membrane. So now ETC will try to work harder to create that proton gradient that's being lost. So ETC actually starts running faster whenever we have an uncoupler, and it will consume more oxygen. So it will try to run faster, and what it does is just it will produce a lot of heat because it's running really fast, but we're not making the proton gradient that we need to generate enough ATP. So what this does is it creates a lot of heat. So there's tissue called brown fat, and what brown fat has is this protein called mitochondrial uncoupling protein, and what this does is it just uncouples the gradient, and it allows the person who has the brown fat tissue to start burning fat, or like produce a lot of heat and burn fat. So they actually tried to use this as a treatment. They gave people something really similar to mitochondrial uncoupling protein. They gave them uncoupling proteins, and it didn't. They tried to use that as treatment for obesity, and it didn't work out too well because it produced too much heat, and it can actually like cook your organs because of the amount of heat it will produce. This is an example of an uncoupler. Basically, they accept the proton from the cytosol or the intermembrane space, because protons can shuttle between these two freely, so they can accept a proton from the intermembrane space, and then they can go back in the matrix and disrupt the gradient. Okay. Okay, so this, I added the slide as a mitochondrial inhibitors. I'm not going to go through all of them, but you guys are, I don't know if you expected to know them by name, but just know that you can have inhibitors acting on complex 1, 3, 4. I don't know if there's anyone's out there that can inhibit complex 2, but the ones listed here inhibit complex 1 through 4, and then some of them are uncouplers. So these two are uncouplers, and then oligomycin inhibits ATP synthase. Okay. So I added one mitochondrial disease because I thought this one was really interesting. So it's called Leber's hereditary optic neuropathy, and you have deficits in complex 1. The proteins that are the proteins that are affected are ND1, ND4, ND6, the subunits, I mean, and they each have lower activity. The pathologies for this are mainly observed in the nervous system, and this results in the degeneration of retinal ganglion cells. And what happens is that because you degenerate your retinal ganglionic cells, is you have this onset blindness starting from age 7, and eventually they'll have impaired speech and cognitive deficits. So you have blindness because complex 1 is not functioning properly, and then eventually you'll get impaired speech and then cognitive deficits. And then mitochondrial gene therapy can have shown to be helpful in some patients, so you can use that to help treat it. Okay, so in summary, we transferred electrons from complex 1 to complex 4. NADH entered at complex 1. FADH2 enters at complex 2. We did the Q cycle in complex 3, and then the electrons were given the cytochrome C. And then cytochrome C gave it the oxygen in complex 4. Another thing I added, so this is, I keep spelling these wrong. So if you inhibit complex 1, you can still run ETC from complex 2. So in any of these, so when we add these two that inhibit complex 1, we can still run electron transport chain from complex 2. Proton motive force, the gradient that we make, is what drives ATP synthesis. So I talked about that, the F now and F1. And then uncoupling of ETC and ATP synthase happens as a respiratory control mechanism to stop the cell from hydrolyzing its own ATP. So I'll upload this, and if you watch it till the end, I hope you have a great day. If you have any questions, just email me, or you can come to my office hours. Thanks so much for watching.